Yeah, I might just do a little compile like this week. There's some testing stuff. But even just a teeny bit,
Okay, so we left off talking about the in-class practice problems. You guys remember that? And so the question is, what are the odds of getting a purple flower from this cross? Big P, little p with big P, little p.
3 to 1.
So, yeah, so 3, remember, since it's 3 to 1, that's 3 fourths to 1/4, so the probability would be, or the odds would be 75%. What are the odds of getting the big P, little p genotype?
50.
So remember the genotypic averages are 1 to 2 to 1. So the 1 is homozygous dominant, 2 is heterozygous, 1 is homozygous recessive. So what's the answer? 50%, right? 2 out of 4. What about the little p genotype? 1 out of 4, right? 1 to 2 to 1 ratio. So on the test, you could do a bunch of Punnett squares, or you could memorize the ratios. It's up to you. What kind of cross is this, by the way? What do we call it when it's between two heterozygotes? It's a mono hybrid cross. Excellent. So what is this cross? They're heterozygous for 2 traits. What do we call that? Dihybrid. Excellent. So what are the odds of getting a wrinkle green seed from a dihybrid cross? What's dominant here? What's that R? Round, and this one is yellow. So wrinkled and green, they're both recessive. So what's the probability of getting both recessives? Remember the ratio is 9 to 3 to 3 to 1? So it's 1/16. That's exactly right. What are the odds of getting a round green seed phenotype? 3/16, right? Because remember the 9 is both dominant traits, 3 is 1 dominant, 1 recessive, and the other 3 is the flip-flop of that, and then the last one is both recessive. And then the last question is, what are the odds of getting this genotype, big R, and big Y, big Y? Remember, because of independent assortment, we can take these independently. So what's the probability if we do just this one trait, we would get this outcome? What's your genotypic ratio for, what's that cross called? Monohybrid. What's the genotypic ratio? That's the phenotype. So what's that one then?
25%.
Yeah, so 1/4. What's the odds of getting big Y from that cross?
1/4.
So what's 1/4 times 1/4? So that's the probability, 1/16. You guys get it? All right. So we talked about things that follow Mendel's ratios, things that are completely dominant and recessive, but in real life, things aren't always like that. So in the pea plants, remember, white is the absence of color. It's the default color. It's like moving into a new apartment or a house. What's the color of the walls? White, right? There's no paint or anything involved in that. But in some cases, there's really a white paint, right? So if you went to Home Depot and you bought a gallon of red and a gallon of white and you mixed it together and you put it on your wall, what would you get? Pink. So snapdragons are an example of that, where there really is a white pigment. So this is red, this one's white. When they make heterozygous, it's mixing the two colors together because they're both pigments. And what do you get? Pink. So this changes the expected phenotypic ratio. If it was a complete dominance, then you would expect it to be what? Okay, let me back up. Let's do so this would be red, right? So if we do the monohybrid cross, we cross two pinks. What would you expect this outcome ratio to be of their offspring? The phenotype for the monohybrid cross is what?
1 to 1.
The phenotype.
3 to 1.
3 to 1. So you would expect if this was in line with Mendel ratios, there would be how many red and how many white? 1. But it doesn't follow that because this is in what we call incomplete dominance, where the traits are blended together. And so the genotypic ratio is the same as the phenotypic ratio. So what's the phenotypic ratio in an incomplete dominant cross? One to 2 to 1, right? 1 is red, 2 are pink, 1 is white. Right? So on the test, I might say you cross 2 tribbles with each other, and one was red and one was blue, and their offspring were purple. And to test that, you cross the 2 purple offspring, and you got 1 blue, 2 purple, and 1 red. What's that an example of? Really? So we have 2 animals, guinea pigs, whatever, tribbles. One's red, one's blue. You do a cross, and their offspring are all purple, right? Red and blue make purple. You self them, so you cross 2 purples in that generation to each other, and you get 1 red, 2 purple, 1 blue. What is that an example of? Incomplete dominance, right? What if I said you crossed them and you got a 3 to 1 ratio? What would that be an example of? Complete dominance. So you need to be able to associate the ratios to what's going on genetically. Does that make sense? Okay. So there's other variations from Mendel's rules. One of those is you have something called codominance. So codominance may seem like incomplete dominance, but it's really not, because incomplete dominance is a mixture of things. Codominance means that they're both expressed simultaneously. So if we went back to the snapdragons, they wouldn't be a mixture of red and white, right? If you looked at them closely, there would be red and white cells that alternated, and from afar they would look pink. But that's not what happens, right? Each one of the cells are pink. Does that make sense? Here, both of the alleles are expressed. So we have a blood cell.
My cat is that way.
What?
My cat is that way.
So we have a cell, right? And we have 2 alleles for blood groups. And there's actually, this is an example of multiple alleles. You guys probably know about blood groups. So what's the different blood types? O and A and I can't hear you guys, you're so quiet.
We have A positive.
Forget positive and negative. I'm just asking you the blood type. B, AB. Any others? So you guys know that you have 2 alleles for each trait. So what are the alleles for O? All right, I'll give you a hint. So if you have an allele on this chromosome that makes A, it makes a receptor on the surface that's shaped like A. So I'll just make it a sort of a one that fits a circle, like a ball. You could have a B receptor, a B allele that makes a B receptor, and we'll just make that fit a square. And then both of these could be messed up, and then you would make nothing. So that's what O is. O is, it's not making A or B. Does that make sense? So let's go back to this. What's the genotype of someone that has a phenotype of O blood? What's that allele have to be? Well, remember there's A, B, or O. There's only three.
Yes.
So what's that one?
O.
O. And that one?
O.
So that's the genotype. What's the genotype possibility for A? That one could be A and that one could be
A.
O. That's right. Or that one could be A and that one could be
A.
No, that would make you AB blood.
A.
So if, let's say A made three of these, right? You would still have three A-shaped receptors on the surface, right? So the reason your blood type's A is because you have B cells, and those make antibodies, and antibodies attach to antigens, which is what this is. That stands for antibody generator. So there's a B cell over here making random shapes that it'll randomly make one. So it's making, there's millions and millions of B cells in your body, and each one of those are making a random shape. Just on the off chance, it would be like you building Legos, just randomly building them in case you made one that fit into, I don't know, a lock or something, let's say. So this one fits this receptor, so what would we call that antibody? Any ideas? It would be anti for what? If it bound to this. Well, what's that called? No, that letter right there. So it would be anti. There you go. Now, if that binds to this, that sets it up to get attacked by white blood cells. Would you want your blood to get attacked by white blood cells? Then you wouldn't want to make anti-A if you had an A blood type. What would you want to make?
Anti-B.
Yeah. You'd make anti-B because that would protect you from something that's foreign. Does that make sense? All right. So if you had one A allele or two, then you'd end up with 6 of these, right? Would that matter what your your B cells made? It would still make what? Anti-B? Are you guys lost? Okay, let's back up. Here's our blood cell, right? We have an allele, that's a piece of DNA that codes for a messenger RNA that makes a protein that is an integral membrane protein. It goes across the membrane and has a specific shape, right? So if you have one, let's say if this is A and that's A. This one, A, just hypothetically, let's say it makes 3 receptors. If you have 2 of them, it's going to make twice as many receptors, so it'll be 6, right? But does it matter if you have 3 or if you have 6? to the B cell,
No.
it's still going to make
Anti-B.
anti-B, right? You wouldn't want to make anti-A even if you only had 3 because it would still kill your cell. 6, 3, doesn't matter. So that's why your blood type is considered to be A if you're AO or AA, right? Now, if you're B, then you could have the same deal, right? Different shape. And this would have the B shape. So one of these could be B and that could be O, so you make 3 of these, right? If you had 2 B's, you'd make 6. But would it matter to the B cell? What would the B cell make then? Anti-B?
Anti-A.
Yeah.
So is there no anti-O?
So this could be BO or BB, and it would still make anti-A. You guys get it? All right. So you're right, but you're a spoiler. So give me a second to get to that. So you could have one that made A and one that made B, and half of yours would be shaped like A's, and the other half would be shaped like B's. So what would you want to make then? Well, O is nothing, right? That'd be like making anti-air.
But you don't want to kill any of them.
Right, so you wouldn't make any antibodies, right? Because if you made an A or a B, it would kill the cell. If you made an A, it would attack the A's. It's still on the cell, right? If you made a B, it would attack the cell, so it would kill the cell.
So your body doesn't consider anything foreign?
That's right. So if you have AB blood, and let's say you get run over by a car and you're bleeding out, you could just grab any random stranger off the street and take blood from them. Why?
Because it doesn't consider anything
You
foreign.
don't have antibodies.
You wouldn't make any antibodies against anybody else's blood. So you're what they call a universal acceptor. You can take anybody's blood. It's good to be AB, right? And then what's the last one? O? So O is, you can only get OO. Does that make sense? So what would you make here then?
Anti-B and anti-A.
You would make anti-A and anti-B because O's don't have anything on their surface. So you would make both anti-A, that couldn't attach to anything, right? There's nothing there. And you would make anti-B, again, because it couldn't attach to anything, because there's nothing there. Does that make sense?
So they can only take from O.
Yeah, so if you're O, it's bad for you. You can only have O blood. But it's good for everyone else because guess what? You can donate to everyone because you don't have any, it doesn't matter what antibodies people make. It can't attack O blood. So you can donate to anyone. So this is called the universal donor. And if you guys donated blood and you happen to be O, they'll call you. You could have just got home and be like, I just gave, come on down and give another pint. We need your blood because they could use your blood for anyone. And this is the acceptor or recipient, right? So AB, you can take anybody's blood. Does that make sense? So this is an example of multiple alleles because we have more than 2. We've only been talking about 2, right? Round, wrinkled, yellow, green, purple, white. Now we threw another one in the mix. And if you do like hair color, you could be like red allele, blonde allele, brown allele, black allele. So that's what we mean by multiple alleles. And then this is codominance because if you have A and B, it doesn't mix the 2 together, right? You end up with A and B being expressed simultaneously. Does that make sense? Which makes it different than incomplete dominance, because those are mixed together. Any questions about this one?
I have one question. Is it like multiple points on the allele, or is it multiple alleles? I kind of get confused with that. Like,
So-
will it be 3 alleles?
Yeah, so there's 3 alleles. You could have a gene on here that makes the A protein, or a gene on here that makes the B protein, or a gene on here that makes no protein, right? If it makes none, it's still considered an allele, right? Remember the purple white flowers? We still considered white an allele even though it didn't make anything, right? So that's what we mean by alleles. And remember, the gene is for your blood type, your ABO blood type is what we call it. But the alleles are the individual pieces of DNA that make the proteins. And then most students ask about the plus minus thing, so let's do that. So there's another one. It has a different shape. I'll make this a square, a triangle shape. So this one is called the RH factor, and that stands for rhesus. So can you guess where they first discovered this in?
Reese's peanut butter cup.
No, it's not Reese's peanut butter cups. What else? It's named Reese's. Monkeys. So that was first discovered in rhesus monkeys, and so if you have the allele that makes that, what do you think your blood type is?
Positive.
That's right. So you would be Rh positive. And if you don't have the allele to make it, what are you?
Negative.
Right.
Now, what does that protein do? I mean,
It's
what
just
is it?
it's just another cell surface receptor.
But it
Doesn't do
has-
anything.
So it doesn't affect whether or not you can take a.
It does, because just like the A and the B, right? If you have that receptor, would you make antibodies for it?
No.
So then if you're Rh positive, you don't make Rh antibodies. But if you're Rh negative, what do you make?
You make RH.
Antibodies. So if you're making, let's say you're negative and you're making Rh antibodies, you couldn't take someone's blood that had Rh on it, because it would attack it and kill it, right?
Right.
So if you're negative, you can't take a positive.
So like AB.
But if you're
negative-
positive, you don't make any, right?
Right.
So then you can take negative blood. Does that make sense?
AB negative can take only negative then?
Right. So no, you would have to be AB positive to take anyone's blood, because you would make antibodies for. I'm sorry.
Nothing.
Right. You would make antibodies for nothing. You wouldn't make A antibodies, you wouldn't make B antibodies, and you wouldn't make Rh antibodies. But if you were AB negative, you would make
Antibodies for the RH factor.
you would make Rh antibodies, and so that means you couldn't take someone that had a positive Rh, because that would attack those. So if you're AB negative, you couldn't take AB positive blood.
So that's,
But if you're AB positive, you could take AB negative blood. That makes sense?
and that's the test that they do when you're pregnant, right?
Right, because there's certain incompatibilities. If you had a child that was negative, it would make antibodies to your own blood. And if you were negative, and your child was positive, you would make antibodies to their blood. Does that make sense? Okay. So another genetic term that you're just going to have to memorize is called pleiotropy, and that just means that one genetic mess up, one mutation in one allele causes multiple phenotypes. So sickle cell anemia is a good example. It's caused by a single mutation in hemoglobin, but that creates multiple phenotypic effects. So you can end up with brain damage, heart failure, anemia, paralysis, kidney failure, spleen damage, because these cells have a sickle shape, right? So they're not nice and round like wheels. Imagine a sickle wheel. How far would that roll around in your blood vessels? And then the sickle gets stuck kind of like a piece of glass in your vessel, so it's really painful. It's like having a bunch of ground glass going around in your blood. And those they don't roll around very well when they pass through the spleen and stuff, so that gets all blocked up. They don't carry oxygen, so it causes anemia. So you get all these phenotypes from a single genotype, and that's what pleiotropy means. So on the test, I'll probably throw out the definition. I'll be like, what is pleiotropy? And it's real simple: multiple phenotypes from one genotype. All right. So that brings us to epistasis. We talked a little bit about this. Epistasis simply means that two genes work together to produce a single trait. So the one on the board shows the trait for hair color in mice. And what's going on is that there's two genes. You can think about it as like, let's say you want to paint your wall, right? So you need to buy the paint, but you also need someone to put the paint on the wall. So there's a B gene, and that's responsible for making the paint, and that works with the C gene, which is the painter. So if you have paint but no painter, what happens to your walls? They're white, right? So you need the paint and the painter. So in mice, and this is also true for labs, right? So what flavor of labs come in? Chocolate and vanilla and strawberry. No, just kidding. So brown, black, and yellow. Same in mice, except mice in labs, their default is yellow. In mice, their default is more white. But it's the same phenomenon. So the big B allele produces black paint, and the little b allele produces brown paint. And this big C is for a painter, and little c is no painter, which is basically just another cell that's job is to put the pigment into the hair. Does that make sense? All right. So this is a dihybrid cross, right? These mice are big B, big C, little c times big B, big C, little c. What would our expected phenotypic ratio be if this was straight up Mendel genetics, not epistasis? What would we expect? 9 to 3 to 3 to 1, right? So we would expect 9 with the both dominant traits, 3 with one dominant, one recessive, 3 with one recessive, one dominant, and 1 with both recessive traits. But in this case, you usually get these blend together. So you get a 9 to 3 to 4 ratio, or in some cases, all 3 of these would blend together. So you would get a 9 to, anyone? Bueller? Yeah. So on a test, I might say you cross this and that, and you get a 9 to 3 to 4 ratio. What's that an example of? Epistasis. Yeah, exactly. Or 9 to 7 ratio. What's that an example of? Epistasis. So let's go over it. This one has 2 big B's, so what color is the paint? Black. And if we have 2 painters, so what color is the mouse? Black. This one is big B, little b, so what color is that? Black. And we have 2 painters, so the mouse is black. This one is little b, so what color is the paint? Brown. We have two painters, so it's brown. This one, it's big B, so what color is the pigment? The paint, it's black. No painters. Mouse is white. This one, what color is the paint? No painters. This one, what color is the paint? No painters. White. So we get a 9 black, 3 brown, and 4 white. That tells you that two genes are working together to produce a product. So you could have two black labs that are heterozygous, and you would expect you should get a 9 to 3 to 4 ratio of black to, how many black ones should you get if they had 16 puppies? 9 black. How many brown ones? 3. And how many yellow ones? 4. Any questions about that?
So when you have the black mouse and brown, can they produce the white?
A black and a brown one?
Uh-huh.
Yeah, sure. So you could have, this mouse would be black, right?
Right.
And this mouse would be brown. What did you ask? Could they produce what?
A white mouse.
Yeah, so let's say that this would make a B, right? So the possible gametes are B, big C. Big B and big C, right?
Right.
Or it could make big B, little c. And this one could make little b, big C, right? Or little b, little c. So what would happen if these two got together?
Because they're white mouse.
The pigment is black. No painter. What color is that mouse?
White.
White. Okay. So that's epistasis. The next deviation from Mendel is what that can happen is when you have polygenic inheritance. So what's poly mean? Where do you think genic comes from? Genes, right? So polygenic inheritance means that multiple genes control a trait. So in humans, geneticists know that there's at least 3 genes that control a trait. So how many alleles are involved in this? How many alleles per gene? 2, right? So there's 6 alleles. And you can get all possible combinations. So for every allele you have, you make a certain amount of pigment. So if I have 1 allele that makes a pigment and 5 that don't, then let's go through the spectrum. I have 6 alleles and none of those make a pigment. What color is my skin?
White.
White, right? If I make 1 unit of pigment, it's a little bit darker, right? 2 units, a little bit darker. 3, 4, 5, 6. So you guys get it? The more alleles you have for that pigment, the more pigment you make, the darker you are. And this is a trihybrid cross, so how many gametes can this make? Remember the rule is the number of alleles per gene and how many alleles there are. So it's 2 to the 3rd, which is 2 times 2 is 4 times 2 is 8. So you would make 8 gametes, each parent. So that means when you do a Punnett square, you have an 8 by 8 Punnett square. So how many squares would you have? 64. So that's why there's 64 probabilities here. So you have a 1 in 64 chance if you're heterozygous for all 3 of these to have a child that's completely white. And if you're heterozygous, you have a 1 in 64 chance of having a child that's completely dark. Does that make sense? And this has happened. So there was a couple in England that had twins. They're not identical twins. They're fraternal twins. Come on, internet. Thank you. I have the same issue in lab. I really strongly suspect that they're throttling MCC's data. Come on, there we go. So here's the parents, right? And then here's the children. Yeah. So, like, what's the first thing that the guys say whenever they're on Jerry Springer or Maury?
My baby.
Why?
Because she's just not the right color.
Yeah, or it doesn't look like me. But that's not always the case, right? Because sometimes they open up the envelope and they go, "You are the baby's daddy," and that's how it can happen, right?
That's fun.
So there's a more modern. This is another incident, right? Of that. So it's rare, but it can happen.
Oh.
And that's due to polygenic inheritance. So on the test, I might ask you, what's an example of polygenic inheritance? What's your answer? No, it's skin color in humans. All right, so I asked you guys to do a pedigree for the exam. So if you didn't do it, you're gonna have to learn it anyway. Sorry. If you did do it, you're probably already ahead of the curve. What do the square symbols mean?
Male.
Male circles. If it's a dot in it,
Carrier.
and what does carrier mean?
So they have but it's not expressed.
So what do we call that?
It's genotype but not phenotype.
Right, so what do we call the genotype that's a carrier?
Heterozygote.
Heterozygous, excellent. If it's filled in, then what is it? Homozygous,
Dominant.
no, the other one.
Recessive.
Yeah, if it's filled in, then you have the trait, right? Does that make sense? Because if you're a heterozygote, like if you're a female and you have factor VIII, one that works, and factor VIII, one that doesn't, what do you show?
That's like
Carry.
phenotype.
So you show the phenotype of factor VIII. So which one's recessive? To have it or not have it?
To not have it.
Right. So if you're filled in, that means that you don't have it on either chromosome. So what is that?
Oh,
Homozygous
recessive.
recessive. Make sense? Okay. So we already went over this. Just to remind you, what's that? male. What's this?
Carrier female.
Carrier female. What does these two lines mean?
Cousin related.
Right, they're related. Inbreeding. What's this person?
Dead.
Yes.
Oh, no, it's
Homozygous.
Right, so they have the disease hemophilia, right? And this one, male. What's that mean? No. You don't have to be married to have kids.
I mean, father or mother. Really.
Yeah, but the double line. This is normal. This is not.
Cousin.
Yeah. Okay. And then we talked a little bit about this in lab.
Yeah.
Although I couldn't get the slide to come up, so remember what I told you, widow's peak is dominant. It looks like that. And then this is recessive. And we did the genetic testing on this. So this is just an example of a pedigree. You go to the doctor's office and they ask you what your family history is. What they're really trying to get at, they're not really creating a pedigree because most doctors don't know genetics, so they don't really know how to do a pedigree. But they're trying to get at what they know, that this sort of stuff runs in families, right? So if you're, if you had a parent that had, colon cancer, you might want to get screened before you're 50, like, most people. Or if you have a history of breast cancer, right, you might want to get screened for the breast cancer gene and so on and so forth. So that's what they're getting at. But you could find out what your probability is of having it based off of a pedigree. Right? The only way to really find out is to do genetic testing. Okay, so for the final exam, I'm going to ask you guys two questions. It's going to be based on this pedigree, which is of these people. Their last name is Fugates, and they're from a area called Troublesome Creek in Kentucky, and they have a genetic defect that causes them to not have a certain enzyme that makes you have a normal skin color. So their skin appears to be blue. Right? So what does that look like?
That's one silver.
Yeah, so you could take colloidal silver and turn yourself blue. But this is a person right here that did not take colloidal silver. This is what the skin color of the Fugates looks like. In fact, this person is a relative of the Fugates. This is what a person that takes colloidal silver looks like. This is not pink. That's really his skin color. But it has the same effect. I don't know why you'd want to be that, but, and believe it or not, that way to undo that is to take methylene blue. So you take a blue dye and it makes you not blue. Interestingly. So the So this website has the pedigree for the Fugates on it, and it gives a history of it, so you guys can read this if you want. Here is their pedigree. So what's going on here? Why is that allele fixed in their family? How many incidents of inbreeding do you see?
1, 2, 3, 4, 5, 6, 7, 8, 9.
So quite a lot of inbreeding going on in here. You got everybody see that? So these are cousins. Cousins. trying to find a brother and sister in here. I think there's one somewhere. This one. Anyway, so you can see that inbreeding is bad. Hopefully. And so what I want you guys to do, if you want extra credit on the final exam, there'll be 2 questions. One question will ask you, what is the name of the enzyme that causes people to be blue, that's defective? And the second question is, what does the enzyme do? So you can go to Wikipedia. And I'm going to shut off the recording so that you guys, well, I guess the video will still be on here though. But you can read through here, so I'm not going to point it out as a bonus for you guys showing up to the class today, but if you go to the link for, this is the condition. So, what does that say? There's the answer to one question.
Is that a 1?
Yes. And what it does is, so what does the enzyme do? It reduces methanoglobin to form hemoglobin. Alright, so each of those questions will be worth 3 extra
Do they die
points.
early?
No. Alright, so that's all I'm going to say. I'm releasing the audio now. I'm closing this so that we can keep it our little secret, our little extra credit secret. Don't tell anyone. Alright. So that's the end of chapter 14. Do you guys have any questions about that?
So we write it down this 2 question and we give it to you?
No, it'll be on the test. You just got to pick it out of a multiple choice question.
Oh.
Okay, so this chapter is about how our DNA gets replicated and passed on. So we've talked a little bit about the DNA gets replicated, and that's a magical process done by leprechauns and unicorns inside the cell. But really what's going on, you guys know that the workers are enzymes, and so we're going to kind of explore how we figured out DNA was the genetic material, how we know how it replicates in this chapter. So the first part is, how do we know that DNA, when did we figure out that DNA was the genetic material? So in the 1930s, scientists knew that about chromosomes, right? And what are chromosomes made out of? DNA and histones, right? Protein. And so most scientists believe that the genetic material in the chromosomes were the proteins, not the DNA. Why do you think that is?
Because they're responsible for making proteins.
Well, they didn't know that, right? So how many letters are in the code for DNA? How many letters are in the DNA alphabet? How many letters are in the protein alphabet?
22.
There's 20. So what do you think you can make a more complex language with? So most scientists believe that DNA, with its 4 puny letters, could never make something like a human being. So they completely disregarded DNA. They thought it was just junk. They didn't think that it had anything to do with heredity. So everybody was on the protein bandwagon at this point. In 1926, a guy named Griffith did an experiment. It's a weird experiment, but you're going to have to kind of follow along with this. So he was doing research on a bacteria called Streptococcus pneumoniae. Can you guys guess what that causes? Yeah, it doesn't cause strep throat, it causes pneumonia. And back in the 20s, what would happen if you got pneumonia? You died. So there's 2 kinds of bacteria, two strains of Streptococcus pneumoniae. One is it makes this extracellular matrix, and we talked about this, right? So the cell can make junk to put outside of the cell. This is made out of a type of material called a glucosamidoglycan. I don't care that you know that. Just know that it makes this big round thing. Remember, just like our blood cells, bacteria have integral membrane proteins. And your body makes antibodies for that would attach to it and attack it. But what's in the way? This big capsule that's made out of proteins and sugars and junk like that. So if you got this form, and this is called the S form because under the microscope it looks smooth, it's round. So S, which stands for smooth. If you got the smooth variety of Streptococcus pneumoniae, what do you think happened to you? You're dead because your immune system can't deal with it. There's another form that the capsid isn't made correctly, and so it looks like this. And so the antibodies now can recognize and bind to it. So if you got this form, and by the way, this is called the R because it looks rough under the microscope. What do you think happened to you?
You fought it off.
Yeah, you fight it off and you live. So this one kills you, this one you're okay with. So what Griffith did was he did experiments with mice. He took living S strains and he injected it into mice. What do you think would happen? They die, and you're right, they died. He took living forms of these and injected it into mice. And what do you think would happen? We just said, well,
They didn't die.
yeah, they didn't die, they lived, right? Because the immune system, the mouse's immune system fought it off. Then he took these and he heat killed them. So you guys watch like survivor shows where you can like boil water for 10 minutes and you kill all the bacteria in it. So he did that. He injected the dead S strain into mice, and what do you think happened? They lived, right? And then he did something weird. He took the dead ones, S strain, he mixed them with the live R strain, and he injected them into mice. What do you think would happen?
They died.
Why would you think that? If you're going to make a hypothesis on this dead one, you inject it into mice, what happens? They live. This live one, you inject it into mice, what happens? They live. You mix them together, inject it into mice, what should they do?
They should live, but
They
died.
should live, right?
Yeah.
But they didn't, they died. So why?
There's information. They didn't.
Yeah, so there's some possibilities, right? One of those is that the dead one somehow gave something to the live R's that made them, because when he isolated these out of the blood, he found living S1s, right? So the other option is that the dead S1s somehow were resurrected in the presence of the live R strain. So you think that happened? No. So this is the first experiment that it showed that genetic material could be transferred from one bacteria to another. And we call this transformation, and it's still, this term is still used today. So if I took a gene out of a human and put it, they've done this for insulin. So they took the gene for insulin out of a human being, put it in a bacteria, and now that bacteria makes human insulin. So that process is called transformation. And it's this guy, Griffith, that coined that term. So transforming. But we still didn't know what the genetic material was. So it took a lot of other experiments to figure that out. In 1952, just to give you a reference, right? So World War II is over. We've already dropped 2 atomic bombs on Japan. But we still don't know what our genetic material is. So these guys, Hershey and Chase, they did an experiment where they used a virus called a T-even virus. So it's T2 or T4 or T6 or T8, right? And there's other T viruses that are odds. So can you guess what their numbers are? 1, 3, 5. Yeah, you get it. So they took one of these called T2, and viruses are real specific for the host that they infect. So these T phages, that phage is a fancy word for virus. T phages infect E. coli. So we have an E. coli sitting around minding its own business, right, in your colon or wherever. This virus comes along, and it looks like a little spaceship. Inside, it has DNA. And remember, viruses are different than eukaryotes. They're similar to bacteria. So do bacteria have histones? No. So their DNA is just DNA. There's no proteins mixed in with it. Right? So that's what makes it unique. This capsule is made out of protein. So scientists really didn't know what these viruses were injecting into the E. coli, but they knew they were injecting something in there. And whatever that was had to be the genetic material, because it made more viruses, made lots of viruses until the bacteria exploded and the viruses went all over the place. So whatever that is that's going into this E. coli cell is the genetic material. So they labeled these by growing the bacteria and the viruses together in a flask with a radioactive element in it. So one of these elements is unique to DNA. It's not found in protein. What element is found in DNA but is not found in protein? I pointed this out in chapter 4.
It's the nucleotide.
So that's a periodic table of elements. What element is unique? It is. It's the phosphates. So DNA contains phosphates, but proteins do not. What is unique to proteins that's not found in DNA? What element?
Nitrogen?
What did you say? Or you gave up on me? You're right, it's sulfur. So they used P32 as a radioactive form of phosphorus and S35, which is a radioactive form of sulfur. They grew the bacteria in the presence of this radioactive material. So that would get incorporated into the DNA as they replicated. And then they centrifuged it. So you guys know when we centrifuge something, we get two phases. What is that?
The pellet.
And the supernatant. So if the viruses were injecting radioactive material inside P32 inside the bacteria, and the DNA was the genetic material, what would be radioactive, the pellet or the supernatant? The pellet. Just like when you guys spun your cells, right? They went to the bottom. So then they did it again using sulfur, and if sulfur was the genetic material and it got injected into there, so that would mean proteins was the genetic material, where would the radioactivity be? Well, it would still be in the pellet, right? So the question is, of these two experiments, which pellet had radioactivity? The one in the phosphorus or the one in the sulfur? You guys probably know the answer to this. So here it is, S35. The radioactivity is in what? Right there. In the supernatant. And P32 radioactivity is in. So what's the genetic material? DNA. These are the first people to figure out that DNA was our genetic material from this experiment. So other experiments showed that gametes have half the number of DNA, the amount of DNA, which supports that. Cells going through S phase have how much DNA? Double. So that also supports this. But scientists didn't know what DNA looked like, what the structure was, or how 4 letters could code for such a complex thing as like a human being or whatever. So it was 3 people, actually 5 people that contributed to figuring out the structure of DNA. Chargaff was a chemist, and he did a lot of research looking at the nucleotides. And what he found was that no matter what organism he looked at, let's say if the percent A was 10%, what percent would be the T's then? Because A's always pair with T's, right? And if that were true, then what would the G percent be? 90?
Oh,
What that
40.
Right? So because they all have to add up to 100%. Does that make sense? All right. So on the test, if I give you any percent of any nucleotide, you should be able to calculate any other nucleotide, right? So if I say you have an organism that's 5% G, what percent A is it?
45.
So C is 5%, then T is 45%, and A is 45%. Does that make sense? Okay. Then Maurice Wilkins and Rosalind Franklin, their expertise was taking molecules and crystallizing them and then shooting X-rays through it. You can look, it's kind of like taking a weird picture of like a silhouette, so you can kind of tell what an animal is by looking at its shadow. So they could tell what the shape of molecules were based on the shadow they produced from X-rays. But this back in the day, this was in the 50s, the X-ray machines weren't really good, so they shot X-rays all over the place. And so it took like days and days. So you guys know you can go to the dentist now and get an x-ray, they just go, bzzz, and that's it. If you wanted to do that in the 1950s, you'd have to sit there for a long time to get a crystal of a molecule image. And then Watson and Crick, you guys know who that is? Those guys are? This one's still blinking, so I can't turn it on yet. This side of the room. I apologize. So, you guys know who Watson and Crick are? There's a picture of them on the very first page of your chapter 16, Building the Structure of DNA. So those are the guys that were credited for determining the structure of DNA. But they really weren't the ones that did it. So I'm going to tell you a little story. So Chargaff right here, we talked about that. He knew that percent T's equal A's and percent G's equal C's. Rosalind Franklin was the best at what she did. So she was the only person on the earth at the time that could crystallize DNA and get an x-ray picture of it. So this is her photo. This is called Photo 51, right? Not Area 51, because it was the 51st picture that she had taken of the crystals of DNA. And so she took the picture, put it in her lab notebook, wrote in there that she thought that was a double helix, right? Put it in her drawer in her laboratory and locked the drawer. You would think that her information would be safe, right? You would be wrong. So she worked with a guy named Maurice Wilkins, and they worked together. And back in the day, you guys have to remember that this is in the 50s, and so in the scientific in university communities, they had like these sort of country clubs, right? Kind of like the Masters Club is right now. So in the Masters Club, how many women members are there? Do you guys know? Well, there weren't any until Condoleezza Rice got inducted, or the Citadel, right? So back in the university club, no women were allowed. So Rosalind Franklin was excluded from these males that were conspiring against her, which included Maurice Wilkins, Francis Crick, and James Watson. So what happened was these guys would get together, and Maurice Wilkins worked with Rosalind Franklin at a place called King's College. And so Maurice Wilkins said, hey guys, Rosalind Franklin has a picture of the structure of DNA, and I have access to the lab. So what do you think? So he wrote them a note, which is later found. It says, this is written by Maurice Wilkins to Crick. It says, I think you'll be interested to know that our dark lady, I don't know why he called her that, Franklin, leaves us next week. At last the decks are clear and we can put all hands to the pump. It won't be long now. And he signed it in. So based on that note, Watson and Crick got on the train from their college, which was about an hour and a half away, and rode the train to King's College. And Maurice Wilkins opened the door for the lab and let these guys in. And they took her lab notebook out and wrote down all of the data out of it, and even took pictures of the picture. And then a month later, they had developed the structure of DNA, which you can see in your textbook. But they couldn't get it right. So who do you think they called that asked to come and help fix it? Yeah, they called Rosalind Franklin, and she didn't know that they had stolen her work. So she helped them model the structure of DNA. Maurice Wilkins was promised that his name would be on the paper, but guess when they published the paper whose two names were on it? Watson and Crick, and they won the Nobel Prize for that. So Maurice Wilkins complained because he said that he had a hand in developing the structure of DNA. So they gave him a Nobel Prize too. But Rosalind Franklin never got one.
Oh my God.
And the reason is because she died from cancer developing those pictures of the structure of DNA. And they don't give Nobel Prizes to people that have already passed away. So they don't give them posthumously. So I just want to ask your guys' opinion of this. So, in 1994, James Watson gave a commencement speech to Harvard Medical School, and it said, "There's a myth that Francis and I basically stole the structure from the people at King's. I was shown Rosalind Franklin's X-ray photograph, and woo, that was a helix. These are biochemists. They wouldn't know a helix if it walked up and slapped them in the face. A month later, we had the structure. Wilkins never should have shown me the thing. I didn't go in the drawer and steal it. It was shown to me, and I was told the dimensions." What would you call that if you went into somebody's house and took their research paper and wrote it as your own and turned it in to a professor?
Stealing.
So what would you call it if two scientists went to another scientist's lab and took their data and used it and published it without giving them credit for it? What do you call that?
Plagiarism.
It's plagiarism. But these guys got a Nobel Prize. Is that right? So I like to call them Watson and Crick, because that's what we are. So in 1957, the two crooks came up with the model based on the calculations from Franklin. And this is what DNA looks like. So it's a double helix. That was confirmed by her X-ray pictures. We know the size of it from her X-ray pictures. It's 2 nanometers across, which is really small. 3.4 nanometers per turn, and then 0.34 nanometers per base. So how many bases per turn? 10, right? How many hydrogen bonds between A's and T's? 2. How many between G's and C's? 3. And there are proteins that interact with DNA. So there's 2 grooves in DNA. One is called the major groove, this really wide one, and one's called the minor groove, which is this really tiny one. So what do you think where do you think the proteins interact with DNA to read the sequences? The major groove. So the major groove is most important. I won't ask you guys any of that stuff. I'm just giving you sort of tidbits so you kind of get an idea of what scale we're talking about. So the two crooks, they came up with one thing that they've actually contributed to science. And I'm pretty sure my 5-year-old could have done the same thing, just so you know. I'll see if you guys can guess. And then we'll find out if you deserve a Nobel Prize as well. So here's the 3 models of DNA replication. So we have the 2 strands, right? They open up. Okay, 2 strands. They open up. And this one's used as a template. So a new strand is copied onto this template, and a new strand is copied onto that template. Does that make sense? Or you could have the original, come on, DNA, and they would separate, right? And somehow it would copy, and this one would go back to the way it was, and they would make a whole new copy. And so this one is called semi-conservative because it conserves half of the original strand. This one is called conservative because it conserves the entire original strand. And then the last one is dispersive. So you have your original strand that somehow blows itself up and ends up in pieces that look like this. And the new strand would be something like that. And this is called dispersive. So you got 3 guesses. Which one do you think is the way that DNA copies itself? Hey, you all deserve Nobel Prizes. Congratulations. So they proved that. Those guys didn't do the experiments. They just wrote about it in their paper. But what they did was they grew organisms and heavy nitrogen. So what's the nitrogen's molecular mass normally? 14, right? We agree? So you should have learned that in chapter 2. Hopefully you know that. But there's such a thing called nitrogen 15. So what does that mean?
It has an extra neutron.
It has an extra neutron. And so if you copied this with, say the green is the heavy nitrogen, and you ran this through a gel, kind of like we did in lab, how many bands should you get? In other words, would these be the same weight? Well, they should, right? Because this is a mix of heavy and light. This is a mix of heavy and light. So these should be relatively the same weight. This one is light and this one's heavy, so how many bands would you get here? 2. That would be correct. And this one, they're relatively the same, so you should get 1 band, right? So they did that, and you can see here that they didn't get 2 bands, so they could exclude conservative. So this one is out. But they couldn't tell this one from this one. So they had it go through another round of replication. And if it went through another round of replication, the new strand, these would separate, right? So you'd have 1 green and 1 purple. And then the new strand would be completely all heavy, right? And then this one would be half heavy and light. So if this one blew all up, then it would still be, they would all be the same mass, right? So this one should have 1 band, and this one should have 2. 1 mix and 1 all heavy. So they did that, and they got 2 bands, so that excluded this because it should have 1. And so they showed that DNA replication is indeed semi-conservative. So Watson and Crook, thank you for guessing the correct way that DNA is replicated. That's it. So, any questions about anything so far? All right, so we only have 2 more lectures. So the plan is finish up 16 and start 17 on Tuesday, and then finish 17 on Thursday, and then I might have a live review on Monday or something like that, depending on how many people come, because I don't want to be here by myself. Or I could just post an old review online from like a spring semester. So we'll take a poll next week and find out how many people are going to show up. All right. Any questions? Tests aren't graded yet. I have the grades, but I haven't like gone through and calculated, added in extra credit and stuff like that, and recorded the grades. I do know that the average in this class was a little lower than my other class, but look at how many people come to lecture, right? My other class has 48 full seats. So they got about 8% higher on the test than you guys did. So their average was a 73. I think yours was about a 65 or 66. So I may have to curve that. I don't know. We'll see. But they should be posted by tomorrow evening on Canvas. All right. Have a good weekend.
When you were talking at the beginning of class about doing like the study for the new skin and stuff like that, the genealogy or whatever, are we supposed to be reading, wasn't that for this last test?
No, so I don't want you to bring in the genealogy. I'm going to ask you. Let me show everything.